today we'll do TCA cycle after finishing up pyruvate dehydrogenase complex, and then we'll do glycogen metabolism and glycolysis run backwards on Friday, gluconeogenesis pathway. And then we will have hit all the reactions in both directions that we need to look at for the purposes of talking about regulation. So next week we'll have, the whole week will be on regulation, which will mean that you don't do new pathways, but rather we just look at the ones we have already looked at and think about how they're controlled in the cell, how it works. And then we'll go on from there to talk about some aspects of nitrogen metabolism, fats and such. And then we'll be ready to wrap up with oxidative phosphorylation, which is actually shown here, respiratory electron transport chain, which will come up on some other slides today when we talk about TCA cycle, because what TCA cycle does is basically fully oxidize the glucose, the CO2. Okay. So we talked about, last time we talked about, started to talk about how pyruvate is converted to acetyl-CoA in the pyruvate dehydrogenase complex. And we noted that glycolysis occurs in the cytoplasm, but the TCA cycle occurs in the mitochondria. Most of the reducing power, most of the electrons that we peel off of glucose in order to ultimately combine with oxygen, most of those electrons are coming off in the process of the TCA cycle. A little bit came off in the process of glycolysis, and so there was a free energy yield out of glycolysis, but it was only about 5% of what we could get out of glucose generally, of the full glucose molecule. We'll also get a little bit of energy yield out of the pyruvate dehydrogenase complex that we're talking about now, and then we'll get most of it after the TCA cycle. So it's pyruvate that gets translocated, so specific charged compounds do not just move from one cellular compartment to another when they're bounded by organelles and by double membranes. Usually the outer membrane is permeable, which is the case in the mitochondria, but the inner membrane is selective. So pyruvate can sort of poke its nose into the mitochondria by getting through the porous outer membrane, but then it needs to go through the translocase in order to actually get into the inside where the complex that we're studying is. Right? And once it's inside, the electrons that come off of this enzyme in the form of NADH and then come off of the TCA cycle in the form of NADH and FADH2 will find themselves in the right position to kind of go back to the inside of the inner membrane. On that inner membrane is where the ATP synthesis process is going to happen. So the pyruvate dehydrogenase complex, abbreviated either as PDH or PDC, is a monstrous enzyme of about 8 million molecular weight, which makes a big structure that looks like this ball on the upper right of this slide. It has 3 different kinds of enzymes in it. So, and they're organized into this large supramolecular complex. And E1 and E2 and E3 with these sort of enzyme names are enough to do the reaction. It's not like you need to pass the pyruvate around the whole outside of this gigantic thing. Rather, what happens is lots of different pyruvate molecules are coming along and binding different parts of this molecule, and reactions are occurring simultaneously. Right? So, of course, we just need to look at what goes on in one set of E1, E2, E3 molecules, and you can see just from these cartoons, that E1, E2, E3 are going to be localized all over the big ball, but they're spread out so that there's always E1, E2, E3 contact, no matter what site on the ball you are. We talked about channeling, and that occurs over the course of this 5-step reaction. So let's look into the pieces of the reaction. The facilitation of this reaction happens by way of cofactors, and there's 5 of them that we need to know about for this one reaction. some of which you already know about, NAD and FAD, of course, you know about as the redox cofactors. Both of them are involved in this reaction. There's another redox cofactor as well, lipoate. I'll show you the structure of it in a moment. Thymine pyrophosphate is a 2-carbon carrier, and we talked about that with respect to the mechanism of how pyruvate is decarboxylated as one of the fermentation mechanisms. So thymine pyrophosphate is a cofactor that facilitates carbon-carbon bond cleavage by virtue of having a positively charged thiazolium ring. So the missing one that we haven't talked about, CoA is here, coenzyme A. And coenzyme A has all this cabbage on it, right? Which you should be able to recognize it. I wouldn't ask you to draw all this out. But at the end of it is a thiol group. So it will form a thioester. So acetyl-CoA, which is the product of the reaction, pyruvate to acetyl-CoA plus CO2, that's the overall reaction. So we're losing a CO2 here. Right? So on the straight pathway from glucose through glycolysis into TCA cycle, the first CO2 is split off here. Right? And so you could say, well, it's 2 CO2s because glucose made 2 pyruvates, and 2 pyruvates make 2 acetyl-CoAs. So we got 2 CO2s, so we got 1/3 of the glucose carbons are now oxidized. So here, when it says acetyl-CoA, this CoA here is hiding all that stuff. Right? So here's a cutaway view of that big ball. And it shows you, different E1s, E2s, and E3s kind of scattered through here. The lipoate cofactor is actually attached at a point that is within striking range, so to speak, of all 3 subunits, right? Kind of in this little blue connecting region here. And the lipoate cofactor, this is what it's going to look like. It's got this long, flexible arm on it. Okay? So what it actually looks like is this. So it's a covalent modification to an enzyme. It's the E2 enzyme, so it's one of the 3 unique enzymes that are polypeptide chains, you can call them enzymes, that are part of this big particle. So there's a lysine in a very specific position, and it's modified here. You see an amide bond. Right? And then lipoic acid is this string of methylene groups, and then the business end here, where it's either oxidized or reduced. Okay? It's also got a 3rd state in which it can be acetylated, and that is central to how it works. Notice the acetylated form carrying a 2-carbon group. So if you start with pyruvate with 3 carbons and we get rid of CO2, we've got the other 2 that are left, and those are going to turn into acetyl-CoA. But before they turn into acetyl-CoA, they turn into the acetylated form of lipoic acid. Okay. So that's just attached on the molecule. So this is going to be the released substrate after transfer to acetyl-CoA. Right now it's attached to lipoic acid, which itself is attached to E2. So we've got a covalent structure here as part of the mechanism. Okay, so the business end of this molecule is attached on. The business end is this redox part here, and it's going to get successively oxidized and reduced in the course of reactions, so that it's ready for another round. So that's one important aspect of lipoic acid, this part up here with 3 different forms. And the other part is that it's floppy. And the way you know it's floppy is it's all single bonds here. Right? The only place where it's like marginally not floppy is they have clearly some limited rotation around that amide, because you have partial double bond character there, like a peptide. Right? But other than that, it's all floppy. And so what that means is you've got the business end of this thing, which is going to pick up what's going to turn into your substrate at the end of 10 methylene groups worth of floppiness, which extends out for a long way. Right? Carbon-carbon bonds are 1.5 angstroms, so that's a big distance. In fact, it's a big enough distance to span. If you attach here and you imagine that thing could flop this way,